Hello, my name is Joyce Harper. I'm Professor of Human Genetics and Embryology at the Institute for Women's Health, University College London. And as part of Global Women Connected, I'm making a number of vlogs on women's health issues. And this vlog is about genetic inheritance, and it's in a series of vlogs about genetic disease. And in the next vlogs, I'm going to talk about genetic testing. We have 23 pairs of chromosomes, one set we inherit from our mother and one from our father. 22 of these pairs are called autosomes, and the last pair are the sex chromosomes. If we've got two X chromosomes, then we're a female, and if we've got an X and a Y, then we're a male. Our genetic code can be looked on as a series of books. So we've got 23 volumes of these books, each representing a different chromosome. Within these books, we've got loads of chapters, and these chapters represent the genes, and the genes are made up of a series of letters which are actually bases in our genetic code. If there's an error in the genetic code, in these words, in the chapters, this is called a mutation, and this sometimes can lead to a genetic disease. Now, the main genetic disease is called a single gene defect, and this is caused by just an abnormality of a single gene. And there's different ways that single gene defects can be inherited. We can have a dominant single gene defect, where you only need one abnormal copy of the gene to have the disorder. So you would have a 50% chance of passing this abnormality onto your child. And these are often late onset diseases, such as Huntington's disease. The next type of genetic disease is a recessive single gene defect. And here you need to have inherited an abnormal chromosome for that disease from your mother and your father. If both of your copies are abnormal, then you may have the disease. For this disease, you've got a 25% chance of passing this on to your child. These are diseases such as cystic fibrosis and beta thalassemia. Then we've also got diseases where the gene is on the X chromosome. These are called sex or X-linked diseases. The mother will carry this gene. She won't have the abnormality because she's got another X chromosome that is fine, but she will be at risk of passing this disease on to 50% of her boys. Her girls will have a 50% chance of being a carrier. Examples of sex-linked disease are haemophilia and X-linked mental retardation. There are also multifactorial diseases, and these are caused depending on your genetic background, but also affected by the environment, and they can be affected by a number of genes. Examples of this are heart disease, diabetes, and obesity. For the majority of single gene defects, there are many different places within that gene that an error can happen. So, for example, with cystic fibrosis, there's literally thousands of different mutations within that gene that can lead to cystic fibrosis. Now let's talk about the chromosomes. So we have 23 pairs of chromosomes, and we need to have this balanced chromosome arrangement in the fetus, otherwise it will normally not survive. So it could be just a fragment of chromosomes that are extra or missing, or we could have a whole chromosome extra or missing. Most of these abnormalities, these unbalanced chromosomes, are going to be totally incompatible with life. So what will happen is the fetus will miscarry. And we know that in 35% of miscarriages, there's a chromosome abnormality of the fetus. There are a few chromosome abnormalities that are compatible with life. The most obvious one is Down syndrome, and there's an extra copy of chromosome 21. But there can also be abnormalities of the sex chromosomes, such as in Klinefelter's syndrome or Turner's syndrome. Now, unfortunately, as a woman ages, the chromosomes in her eggs become very unbalanced. And this really kicks in around the age of 35, and it's called age-related aneuploidy. And it really becomes significant over 40, and especially over 42. So this leads to an increase in miscarriage because of unbalanced chromosomes during pregnancy, but it also causes a decrease in fertility rate. There are several stages when we can do genetic testing, and I'm going to go through these in the next vlog. These are tests that we can do before a couple decide to have children. We call it preconception testing. We can also do preimplantation genetic diagnosis, which is what I've worked on for the last over 20 years, and I'll talk to you about that. We can do prenatal diagnosis, which is on the pregnancy, when the woman's pregnant, or we can do postnatal diagnosis, which is after delivery. Now, most of these genetic tests would be asked for and organised by your doctor, but now there is something called direct-to-consumer genetic testing, and I'm going to talk about this in the next vlogs. The trouble with direct-to-consumer genetic testing, you can even now buy them on the internet, or even now in some shops, some chemists. The trouble with this is that you don't have the information about what is being tested for and what would be the issues if the test was shown to be positive. And in my view, we need to ensure that we've got sufficient genetic counselling before we do any type of genetic test. The last thing I wanted to say that there's also something called genetic screening. So in some populations, they have a very high prevalence of a particular disease. For example, beta thalassemia is prevalent in certain communities, such as in Cyprus, Sardinia, and other communities. So in these communities, they would offer a preconception genetic screening test for anybody that's in one of these communities to see if they're carrying this genetic disease, so that then the couple can make an informed choice of their reproductive options before they start to have a child. So I hope you've enjoyed this vlog, and if you'd like to know anything more, then please watch the future vlogs, and I hope you enjoy them. Thank you.